Hello guys, welcome back to Biotic World. Today let us understand about the chromatography. Chromatography is a technique of separation of different pigments, especially we are using it for biochemical analysis, right? So, the first application by MS Twaswat in 1903. It is used for the separation of plant pigments. Since the components and different colors, the Greek chromatos for color was used to describe the process, so it means the color writing. Chromatography, chroma means colors, grapher means writing. Okay, so here we can understand the meaning of this word, that is chromatography, right? It is a physical method of separation in which the components to be separated are distributed between two phases. So here we can differentiate our solution, our solute, on the basis of a stationary phase and a mobile phase. A stationary phase and a mobile phase that moves in a definite direction, right? Chromatography is a laboratory technique that separates components within a mixture by using the differential affinities of the component for a mobile medium and for a stationary absorbing medium through which they passes. So in this, what we are doing in chromatography, in this technique, we call it as a laboratory technique that separates components within a mixture by using differential affinities. Affinities refers to the ability of a compound, any element, or the presence of solute in our solution which we want to differentiate. Right, so we have mobile medium which flows from one direction to another, and a stationary that absorb that medium and from where we can detect the presence of medium, right? Here affinity, natural attraction or force between things. The smaller the affinity a molecule has for the stationary phase, the shorter the time spent in a column, right? As we had understood. Next we have the mobile phase which move over or through stationary phase and carries sample along with it, thus resulting in the separation of its component. It is either or liquid, that's known as the mobile phase. Now here we have our stationary medium, the part of the apparatus that does not move with the sample, known as the stationary phase. Here we can see a illustration of chromatography technique. Here is the mixture which we want to separate, and in this experimental way we can differentiate it with the stationary phase and the mobile phase. Here we have three samples we have loaded here, and these moves in this upward direction. So first comes to the here and here and here. Here we can differentiate our mixture in four colors. That one is the blue, black, red and yellow. Okay, so affinity to stationary phase, yellow have the stationary phase affinity, red have, and the black have, whereas the blue doesn't passes, right? Blue is just standing here. And affinity to mobile phase, blue is insoluble in mobile phase. Here we can see the presence of the blue color, right? Next we have black, red and yellow. So black also have the smaller affinity, and red also have larger than the black one, and yellow has the largest affinity, right? So here we have to analyze, identify, purify and quantify our solution. Now let us understand the advantages of chromatography. Chromatography can separate complex mixture with the great precision. Even very small component or the very similar components such as proteins that may only vary by a single amino acid. Okay, so we can also differentiate and also separate those similar molecules which having the only difference of single amino acid. Chromatography can purify basically any soluble or volatile substance if the right absorbent material, carrier fluid, and operating conditions are employed. If conditions or the environment, further, if we know about this thing that if environment is particular to us, so we can live. Similarly, is in the case of every experiment. If conditions are desirable, things will work, right? Next, we have the small material consumption. This is the benefit or the advantage of this chromatography that small material is required. Quantization has a broad linearity range, right? Quantization has a broad linearity range. Analysis of several compounds can be done during one run, right? Chromatography is a fast analysis method, well establishes instrumentation with high level automation is commercially available. So we can do this experiment on a large level. Here we have the different type of chromatography based on stationary phase and the based on mobile phase. Based on stationary phase, we have column chromatography and planar chromatography, whereas based on mobile phase, we have liquid chromatography and gas chromatography. So let's have a quick view towards the kind of chromatography. First, we are talking about liquid chromatography. In this liquid chromatography, we have gel filtration, ion exchange, affinity, adsorption, reverse phase, and metal binding chromatography. Next, we have gas liquid chromatography and the planar chromatography. Planar chromatography also having two types: thin layer chromatography and paper chromatography. Okay. So guys, we will do our next portion in our next video. Thank you so much and stay active with Biotic World.